All right, I'm going to call the meeting to order.
You could sit.
I am going to sit here because we can't get the other camera to go. Hello, everybody online. Be quiet in the back row, please. I'm calling to order the 1248th regular meeting of the Entomological Society of Washington. And if you are not familiar with their history, this is the 140th year of the society. 140th anniversary. I'm going to first call on Secretary Gary Hebel to read the November minutes. Gary, you want to have a seat there or from the podium? Okay. Imagine Gary Hebel speaking. It's not hard.
Okay. The 1247th regular meeting of the Entomological Society of Washington convened at approximately 7:30 p.m. on the 7th of November 2024 in room WG 33 of the National Museum of Natural History. In attendance were 8 members, 7 guests, and 17 virtual visitors. Per the usual format of ESW, President Don Weber requested Recording Secretary Gary Hebel to read the minutes of the annual banquet and the October meeting. After these minutes were read, there were no changes for improvements suggested by the audience. The next item of business was discussion regarding a conversion of the society to the status of a nonprofit corporation. President Weber had passed out a summary sheet of the proposed bylaws changes at the October meeting, and these are here entered for the record. Proposed bylaw main changes. Conversion of Entomological Society of Washington from its status as an unincorporated association into a District of Columbia nonprofit corporation in November 2024. This allows proper management of available funds for investment and tax-exempt status to encourage contributions by members and others to the society. Verifying that we are a member-governed society and keeping the language that the society is organized and operated exclusively for scientific and educational purposes. Stating simplified membership categories of regular student, life, emeritus, and honorary. Explicitly allowing electronic participation for official meetings, adding website administrator to officers and describing duties, clarifying that a past president is also an officer, consistent with status as an incorporated 501c3 corporation. Reserve funds can be managed as investment accounts. The treasurer executes investment plans per finance committee's recommendations and approval by the executive committee. After further discussion, a vote was taken for a change in the bylaws, and a quorum of 21 yes votes, including virtual members, achieved passage. President Weber announced the sad news of the recent death of Society member Donald R. Davis. Manon Davis then offered comments about Don. There was a quickly arranged memorial service for Donald Ray Davis at the Wat Thai Temple in Silver Spring, organized by Vishai Malakil. Manon displayed a large color photograph taken by Vishai of all who attended the event. She noted that Don had published over 200 peer-reviewed articles and books, mostly on new species, genera, and families of primitive small moths. She displayed 6 major publications, including 2 on new families. Don had been a member of the Entomological Society of Washington since 1961 and served as president of the society in 1977. President-elect Andrew Jansen read the names proposed as new officers for the society in 2025, provided by the nominating committee. Solita Somoes for President-elect, Abigail Kula for treasurer, Gary Hebel as recording secretary, Alan Norbaum as program chair, Jerry Cook for editor of the proceedings of the society, Matt Buffington as membership and communications secretary, Alyssa Seaman as curator, and Lourdes Chamorro for the new position of website coordinator. Society members will vote for the candidates at the December meeting and are encouraged to nominate other names for the positions. Officially, the following people become new members this evening, having been announced last month as candidates. Evan Economo, Julie Craves, Pamela Zader, David Hewitt, Lauren Wiedner, Thomas Skarlinski, Derek Rosenberger, Andrew Meads, Michael Catarino, Austin Fife, Robert Finn, Alan Cabrero, and Hector Gonzalez Hernandez. Lourdes Chamorro, in her position as acting membership and communications officer, announced Alex Smith Houston as a new applicant for membership. Lourdes also noted that the society now has 142 members. President Weber noted that the ESW meeting in January will occur on the 9th of January rather than the 2nd of January due to a schedule conflict. In new business, Program Chair Alan Norbaum stated that there are current vacancies in the honorary members listing of the society, and that the executive committee will discuss this in a December meeting. Program Chair Alan Norbaum introduced the speaker of the evening, Armando Rosario Lebron from APHIS USDA in Beltsville, whose presentation was titled "The Invasive Threat of Imported Invertebrate Pets." There has been a recent increase in sales of invertebrates online as pets in the United States. APHIS is involved in regulations of movements of live invertebrates across state lines as well as international trade and sales. Millipedes are a widely popular group in sales, but require strict humidity levels as pets. Mantids are quite popular also, but don't live long in captivity. Suppliers of this group gain large profits and have created breeding facilities. Among arachnids, tarantulas are the most popular group of their size and are sold at $75 per animal. Scorpions are popular, and even deadly species can be purchased by mail. Sadly, vendors sell all kinds of ants, charging $50 to $300 for a queen. There is a strong market for insect and arachnid pets, with many vendors in China. In general, youngsters should not be discouraged for their interest in invertebrate pets. Permits for transport should be gained by both buyers and sellers. Too often, children wish to release their invertebrate pets to nature at the end of the season. Such pets should be frozen. International smuggling of invertebrates is a widespread problem and should be reported to authorities. The meeting was adjourned at 8:35 p.m. Questions, suggestions, comments? What names?
Thank you, Gary. If there's no objection, the minutes are adopted. Our next order of business, I will introduce our treasurer in a moment, is to complete the process that we started last month by passing our revised bylaws that call for a conversion of the society to an official nonprofit corporation in the District of Columbia. I would like to again acknowledge that the new bylaws are owing to the long and hard work of Abby Kula, our treasurer, and we need to have a membership vote on this. And Alan, can Abby share the? Should be able to. Abby, can you share the?
I don't think I can share because the current slides are being shared. I don't think it'll let me.
You got
Oh, wait. Oh, maybe it is going to work.
Maybe it is going to work. Okay, here we go.
Is it working?
Yes, it's coming
Excellent.
up.
Okay.
It's got to sync with the, I don't know why it's not syncing locally. Does that take a minute? Alan. It's showing on Zoom, but it's not showing to the audience here. There
We're
you
gonna just
go.
do quick and dirty that way.
All right.
Did it work?
The Zoom
Okay,
interface,
both in
but-
person and Zoom are visible. Abby,
Okay, great.
carry on.
So, like Don said, this is step 2 of our incorporation process we've been working on with the lawyers. And the first one was to approve our new bylaws, which we did last time. And this time they've told us that we need to tell you about and have you vote on this plan of conversion. So it's being adopted today. So that, sorry, I'm trying to get my bearings about me in this document. We are an unincorporated nonprofit association, and this is a plan of conversion that is pursuant to all the codes in DC. And we're converting this into a nonprofit corporation to be known as the Entomological Society of Washington. So we'll be a nonprofit corporation at this point. So all of us, the executive committee members have agreed that this is important to do. And so we're asking you to vote on and agree with these statements here that you are deeming it advisable in the best interest that will be converted to a nonprofit corporation. And that we are desiring as a group to adopt this plan to facilitate and document the conversion, and that we are, by voting on this today, to support it today, we are adopting the name and form prior to conversion. So Entomological Society of Washington, and the name and form after conversion, Entomological Society of Washington. So no change there really. And the effective time should be at the end of the month, end of the year, really, December 31st. And we'll convert this. The previous unincorporated will cease to exist, and then will be this nonprofit corporation.
Abby, if I'm
Yeah, there's a whole bunch of legalese and groups. And then it tells us who the members are, who the directors and officers are. And then here it outlines the articles of incorporation, statement of conversion, bylaws, and then this authorization. So yes, go ahead, Dawn.
Oh, thank you. So we do have a written copy here.
Oh, great.
The form that I need to fill out. What I'm going to do as the ad hoc parliamentarian here, Abby, is to consider what you just presented as a motion. Do we have a second from the floor?
Second.
Second. Okay, Alma. Alma is second. And is there any discussion on the motion to follow through on the change in bylaws by converting us to a District of Columbia nonprofit?
I thought I saw a hand up.
Unmute yourself if you want to speak online. Hearing no discussion, I will call the question and move for a vote. And I'm going to do first our members only in person. All those in favor, raise your hand. And we need a count. All right. You got a count?
13.
Okay, all those online members, please put your hand up. And if you can't figure out how to do that,
Do the raise hand.
do the raised hand thing. And if you can't figure out that, do what Jerry's doing and raise your real hand.
It's under the,
It's
I
under-
can't find it either. So
And is there more, Alan, that we can't see? I hope so.
Yeah.
right.
Yeah, hit the blue button. There's 5 on this screen. Do I do it? Looking for my mouse. Have you got a?
counting on that.
Oh, here we are.
Oh,
We
wait.
got another 10. And I didn't even see Jerry. It was 11. Okay. Good, thank you. Motion passes.
Excellent.
And, Abby, thanks again.
Sure.
Everybody who was involved did a whole lot of work on this. And you'll follow up with Gary in terms of the exact wording that needs to be transmitted to the lawyer.
Yes.
Okay, great. Thanks, everybody.
Sure,
Next.
thank you.
Any comments from the peanut gallery or elsewhere? Okay. The next order of business, and that was a unanimous vote, a 2/3 was required. I wasn't going to say that just in case it was close, right? Okay. I'm going to give my report, and I can speak pretty fast when I need to. The year 2024 was an eventful year for our society, and in part because of what we just did. It's been a great honor and pleasure to serve as your president during the past year, and I've learned a lot about our society and its members. And I also learned, to my great relief, that the bulk of the work is not done by the president, but by other officers and committee members. So sincere thanks to all of you for the many visible and not so visible ways that you have served the society this year. Matt Buffington, who, along with Lourdes Chamorro, is in Nicaragua right now, ends his three-year term of president-elect president, past president. He's really been an essential member of the executive committee, and his institutional memory and creative suggestions have made my job a lot easier. And he's a candidate for election to our slate of officers in 2025, which is an upcoming election we still haven't done. Alan Norbaum served as and is also a candidate to continue very ably as our program chair. Thank you, Alan. He arranged the society's meetings for 2024, held in hybrid format here and via Zoom, including 11 impressive presentations, 4 of which you will hear tonight, beginning with past President Matt Buffington and including also Jeremy Frank, troglodytes, Luke Cressline, mandibular rods, Rebecca Clement, aphids with song accompaniment, Karen Burkhart, tree food webs, Yelena Pacheco, fireflies. Have I mispronounced somebody's name badly? And Armando Rosario Lebron on invasive pets. And again, 4 more tonight. President-elect Andrew Jansen planned and coordinated the Society's annual potluck banquet, which is held on Saturday, June 8th at the USDA ARS Log Lodge on the east side of the Beltsville Agricultural Research Center. The members and guests in attendance enjoyed the fascinating presentation by Dr. Dan Gruner of the University of Maryland, Forest Birds as Apex Predators in Arthropod Food Webs, which transported us back to the Brood 10 cicada emergence and the amazing multi-trophic ecological impacts of the cicada emergence, discovered by Dan's impressive collaborations. Andrew also assisted in assembling the nominations for the 2025 officers, which we will soon be voting on. The auditing committee will, in the future, review the finances of the NSOC of Washington under our new bylaws in cooperation with our treasurer, Abby Kula, who has been, as you heard, quite occupied with our bylaws revision. The detailed report is deferred until our next meeting on January 9th. The summary report from our treasurer, which I have in my hands, shows that the NSOC of Washington is in good financial condition. Our general fund grew from $81,650 to $1,696, reflecting net receipts of $63,136 and disbursements of $44,091. Our special publication fund was nearly stable at $93,027. We all owe huge thanks to our new editor, Jerry Cook, who is online, and our past editor, Mark Metz, and the 7 subject editors who assist them for the publication of the proceedings of the Entomological Society of Washington, which is one of our most important functions of the society. The last issue of volume 125 was published on July 3rd, concluding the editorship of Mark Metz. Mark expertly guided the journal since 2020, maintaining the strength of the journal through the COVID pandemic and aiding a seamless transition to the editorship of Gary Cook. We are catching up to date. 3 issues of volume 126 were published this year, including issue 3, which will be published tomorrow. A total of 27 papers, 12 notes, 1 memorial note, 1 book review, and the Society Minutes for 2023. Issue 4 is in progress and will be published early in 2025. In 2024, there were 60 manuscripts submitted to ESW. Of these, 5 were rejected. Sorry about that. And 4 are accepted and will be published soon in issue 2 with the remaining issue 4 with the remaining manuscripts under review. The journal is working also with 2 groups for special issues and also with authors on a monograph as either a miscellaneous publication or memoir of the ESW. The success of these plans is obviously relying on the continued submission of quality papers by our members and colleagues. The editorial team deserves our praise and gratitude for maintaining quality, high standards, which the proceedings are so well known for. We signed a new contract in 2024, effective 3 years, beginning of 2026, with Bio1, which will guarantee our revenue from this publisher, for downloads and subscriptions to the proceedings. This type of revenue already offsets much of our costs for journal production, but the new contract will guarantee revenue at a higher level. This allows our members to publish papers with up to 2 full color plates without page charges, a major benefit for membership. Thanks again, Jerry. Thanks to the tireless efforts of our treasurer, Abby Kula, we have successfully revised our bylaws and most recently voted for the conversion to an official nonprofit in the District of Columbia. And Gary Hubble outlined the basic changes that happened in the bylaws, which include a new officer, a webmaster, or web page. What is it? It's referred to. That's the short name, but the official name, I think, is website coordinator or something. Anyway, we know what it is. All right. Finally, thanks to our webmaster, our ad hoc webmaster, which will then, with 2025, become an official part of the executive committee. Thanks to our webmaster, Lourdes Chamorro, the Society's website has been very stable over the last year, no major downtime or hacking activity, and it now incorporates an increasing number of features, including links to all journal issues, merchandise, recordings of presentations, and many other features. So visit. And Lourdes will report on the website developments at the January 2025 meeting following her return from Nicaragua. Lourdes took over also as acting membership and communications secretary from Elizabeth Young, who resigned mid-year, and she will report on her membership statistics at the January meeting as well. Talita Simoes, am I pronouncing your name right?
Yes.
Say it.
Simoes.
Simoes?
Yes.
Okay. Has served very ably as our curator, cataloging each and every one of our holdings of the ESW proceedings, memoirs, and miscellaneous publications, which total over 6,000 items. She looks forward to developing a plan for accommodating the needed items, developing a longer-term plan, which would include for distribution of surplus volumes, given that almost all of our publications are digitized and now available online. Alyssa Seaman is a candidate for curator in 2025 because Talita is a candidate for president-elect. Last but definitely not least, well, it's not last, it's second to last, but definitely not least, I wish to sincerely thank and recognize our long-term dedicated service of our recording secretary, Gary Huball, who is recording at this very moment. And in the coming year, I look forward to assisting our incoming president, Andrew Jansen. So that's my report. And the next order of business, which I could either call Andrew up to do or just keep blabbering, is the election of our 2025 slate of officers. I will read them out at this moment. And you are entitled, as any member from the floor, in person or online, to nominate yourself or other additional candidates should you wish to at this time after I've read out the current nominees. President-elect Kalita Samois, Recording Secretary Gary F. Hubble, Webmaster Lourdes Chamorro, Treasurer Adam Kula, Membership and Communication Secretary Matt Buffington, Program Chair Alan Norbaugh, Editor Jerry Cook, Curator Alyssa Seaman. And just a reminder, I will become the past president after tonight, and Andrew Jansen will become president for 2025. So we remain as officers, but we're not on the slate. Do we have any motions from the floor in the way of additional candidates or complaints pertaining thereto? I'm hearing you online if you're there. Okay. Hearing none, I'll entertain a motion to approve the slate of officers for 2025. Alan moves it. Do we have a second? Oh, you get the second. Almost, Elise. Okay. All those in favor say aye.
Aye.
And you guys unmute yourself or give some indication. Warren, hello.
Aye.
Indication that you favor this, or later when I say any negatives, you can express that as well. Okay. That were the aye votes. Are there any no votes against the slate? And you may abstain also at this time if you want to indicate that. All right. I'm just trying to go by the rules here, folks. And that passes our slate. Congratulations, all you elected candidates. Ha, ha, ha. All right. Going to the back. We have more elections. We have the election of honorary members and an honorary president. These nominations are presented to you by the executive committee to the membership at large and need membership approval. For honorary president, do you want to fill in for me now? Okay. For honorary president, we have Tom Henry. Should I cite his qualifications, or should we just?
I think you should do it. I think.
Oh, okay. There's some demand for saying that. These are Alan's list. He's Tom Henry has been a long time ESW editor from 1993 to 1995, from 2015 to 2019, and treasurer, and also outstanding career, of course, and many publications in PESW. For honorary members, we have three. David Adamski, who was president in 2013, membership chair 1996 to 97, and led the Young Entomologist Group for many years, of course, published in Proceedings. Secondly, for honorary members, Gary Hubble, who was president in 2008, and has been our recording secretary, let me do the math, for the last 15 years. John Brown is the third candidate for honorary membership, and he was president in 2001, program chair from 2004 to 2006, membership chair in 1998, with, of course, a distinguished career and publications in ESW. Do we have a motion on the floor to approve this group of one honorary president and three honorary members? So moved.
Second.
Oh gosh, okay, record it, Gary. And online, any comments? I'm sorry, Warren, I didn't give you the opportunity to second that motion. He can't hear. Okay. I'll call for any discussion on that. Hearing none, all in favor say aye.
Aye.
All in favor online, either raise your virtual or real hand. And any opposed? Okay, I'll give you a chance to take your hands down if you're not opposed. And okay, they're going down, and Abby's hand is going down in 3 seconds. Okay, the motion passes. Is there, thank you, everybody, and congratulations to our new honorary members and our honorary president.
Tom is online with us.
Tom,
We can all say.
do you want to say, give a short speech? Apparently not. Either that or he can't figure out how to unmute. Sometimes that happens. If you figure out how to unmute later, Tom, you can bring.
Hello?
Yes,
Hello.
Oh, can you hear me?
yes.
Oh, okay. I can't get the video to work for some reason. I have been let's see here. Let me try it again. For some reason it doesn't, but I just want to say I'm very honored, and I thank the executive committee for nominating me for this special position. You know, I've been involved in the Entomological Society since the early 90s, beginning with being treasurer for at least 4 years. And then I became editor for about 5 years. And then there was a lull in the action, and I was subject editor for Hemiptera and so forth all those years, and involved in different committees. And then finally, more recently, I was editor for another 5 years, and I shared it, part of it, with Matt Buffington. We were co-editors before passing on to Mark Metz. And I continue as subject editor and so forth. I believe in this society. It's a wonderful group. We have a great journal, which I've tried to support all these years. So anyway, I thank you very much for this honor, and I hope to continue contributing to the society as long as I can. So thank you very much.
Thunderous applause, Tom, and you're very welcome. You much deserved.
Thank you.
Now, the next order of business is any unfinished business. Okay, that kills that item. I have one item of new business, and maybe you have some others, but as noted by Gary, our next meeting will be Thursday, January 9th, not Thursday, January 2nd. So just make a note of that, and you'll have an announcement. Are there any other items of new business? How about presentation of notes and exhibition of specimens? That includes you online if you can figure out how to do that. Oh my. Well, that's okay, because we have a very full program still, which is the presentation by Alan of 4 speakers, including himself. I'm apologizing to Alan personally by not italicizing the Latin name Anastrepa curvicauda, but, or should it be, maybe it shouldn't be italicized because it's a group. No, I think it should be. And so I'm personally apologizing that that format is really getting to me here. Alan, please step up and introduce our first speaker. You have a list right here. So you can, and I believe Alma is first, even though she's seconded.
1 second.
Yes.
Tom, can I just go quickly back to
Absolutely.
reports?
Yes.
I want to thank Hannah Kaheng, who helped me with the inventory.
Oh, very good.
Counting 6,000
Yes.
books isn't easy, so I appreciate your help.
Thank you. We can insert that in the reports, and the reports will be referred to Gary, and eventually a summary is published in the proceedings, the first, I believe it's the first issue, but I'll leave that to Jerry. Alan,
Second issue.
second issue. Okay, thank you.
Yes, go.
Wow, he's on top of it. I have a full copy of the curator's report, the editor's report, the treasurer's summary, and the program chair's summary, as well as what you just heard me blather about. Alan,
Okay, I was actually gonna suggest that Coden go first.
Okay.
He's online.
go right ahead.
You want to share your screen, Code? Still with us?
I saw.
Maybe he was expecting to go or something. Sorry, Alma.
Oh, are we doing that one then? Is on the content?
Yes, so. Alma. So I thought we'd do something a little different tonight, like instead of having just one speaker give a long presentation, that we could take advantage of, in fact, a lot of our members went to ESA and have is already prepared, if we could share with us again. So first off is Alma Solis. She's a longtime member of SEL, was our research leader for many years, the world experts on the chirality.
You should be able to just tap over and make all of those screens. Just with the arrow keys.
Oh.
So, just make sure. Okay.
So first, I'd like to say that I presented this talk at Phoenix. I could have given any number of talks, but I gave it at Phoenix because this research happened 70 miles south of Phoenix a long time ago. So not moving.
Oh, okay. Gotten confused with the You still have that sharing?
Do you see the
Yes,
screen?
I see it.
It's sharing on right.
So first of all, an expert on Pyraloidea, superfamily, not just Borel, which is the smaller subfamily, family of the group. Feistinea is the largest subfamily of the Borellidae with about 35,100 species. Only 61 species have ever previously been described from Mexico. You think about the size of Mexico, and then only 61 species have ever been described there. It's pretty impressive, or not impressive. They're plant leaf rollers and tires, seed feeders and borers with a few predators on scale insects and relatives. It includes major stored product pests, plant pests, and the biological controlling invasive cactus moths, which you see the picture of the caterpillars on the top right. They vary in wing size from about 4 millimeters to 2 centimeters. So you're going to see the wide gamut. Western Mexico, where we were conducting this research, includes the Tarahumara region, and it remains an underexplored region. It's called the dark spot. And I found this really interesting paper by Ondo et al. The botanists went through and mined all of the herbariums of the world and came up with these dark spots in the world. And so it's the same with insects, probably. It's not just plants. and certainly in the pyraloids. So this was the first known survey for moths in this region. It was funded by CONABIO, the Commission for the Knowledge and Use of Biodiversity in Mexico. We worked out of Yecora, Sonora, and we stay in a hotel, which is about 1,500 to 1,600 meters. Yeah, I don't know what I said about that. I just want to add that we did not mine the collections in the US for Mexican material. So we used mercury vapor lights and sheets. All moths on the sheets were collected. Small moths were pinned and spread in field. Large moths were either spread in field or placed in envelopes and prepared later in the lab. Local high school students from Yecora, Sonora, participated in the study, collected moths to sheet, and learned how to pin and spread moths. There were 10 collecting localities. State altitude in meters and general vegetation are on the chart. Most of them were from the localities were from Sonora. One was from Chihuahua. All are prioritized terrestrial regions of the Tarahumara region. We surveyed them quarterly for 10 nights from March to October during the new moon in 2002. So this shows the different localities, pine forest, cultivated areas, deciduous forest, oak forest, and mixtures of this, like cultivated oak forest, for example, or cultivated deciduous forest. At least one specimen of each was dissected to confirm its identity. More if determined to be new and to be described. These genitalia and antennal line drawings of new species were made by Herb Neunzig, one of the co-authors of this work. So the results were published in the Journal of the Lepidoptera Society in 2023. So we discovered 35 physotyme species, including 3 new species, which you see here. We named 1 species after one of our co-authors, Charistaneus balcazarius Neunzig Luis. This species is named in honor of my colleague, Manuel Balcazar, who was the principal investigator of our proposal to CONABIO to conduct this moth research in northwestern Mexico. He would drive from Colima with collecting equipment, meet me at the Hermosillo airport, and then we would drive east into the Sierra Tarahumara. This Denise on March 19, 2021, was a great loss for the study of Lepidoptera, particularly of moths in Mexico. So some of the results. There were 7 northernmost records from Mexico. 3 of them were cactus feeding. 5 were endemic to Mexico, and 1 was new to Mexico. There are many southernmost records from Mexico. These are things, most of these species were described in the United States. And so these are all just new records. All species were new records for the Mexican states of Sonora and Chihuahua, except Amylois transitella, which is the deadly navel orange worm, big pest in western United States. Its synonym, A. venipars, Dyer, the type localities from Hermosillo. And again, it's a serious prolificous pest in the US. So the conclusions were that our study provided the first baseline for physotyme species that occur in this area. Considering the small geographic area we surveyed, we expect many more species are undiscovered. Almost all physotyme species discovered in the study were new to the states of Sonora and Chihuahua. We expect this to be the case for other pyraloid taxa. Northwestern Mexico is a significant transition zone for pyraloids between the Nearctic and the Neotropical regions, as exhibited by northern and southern records discovered during this study. Future research papers include the remaining subfamilies of the Plurality, the Chrysogyni, and the Apashiani. There are 23 species, 9 are new and 11 genera, and 10 subfamilies of the Chlamydia with 137 species in 80 genera. 46 are new. So I'd like to acknowledge my co-authors, Dr. Herb Noisek, who just died on Halloween this year, and Manuel Balthasar, funding from Conavío, technical support by John Lewis, who used to work for SEL, and Tina Lidwack, who did all of the illustrations. And she just told me today she finished the entire Cramity this year. So then I can move on to the writing part. And I'd like to thank the University of Colima, the dean, the professor from the entomology, and the international specialists. When I found out that he had passed away, I didn't know anybody there. And so I cold called the international office, and they immediately came, set up a Zoom meeting. I explained what we were doing, what Manuel and I were doing, and what the issue was, which is that all of the girdle material is here. There are no export permits, no permits of any sort. And so I had to contact them to try and come up with a way to deal with this. And with Kaylin, we've come up with a way to for them to give us all the material and just return representatives. That's it.
What exactly, when you said cultivated, what exactly does that usually refer to?
Fields.
Like fields?
Fields of crops, basically. And a lot of these, in the paper, it's an annotated list, which talks about its host plants, the host plants of the species, and its full distribution, not just Mexico.
So were you collecting for some of those mountains, like Sky Islands?
This is the king of the video to work.
We are sky islands. From high elevations to lower elevations, we did all elevations possible. The one in Chihuahua in particular was one of the higher ones. And that was where a lot of the new species.
How long was your field season?
A year and a half.
Oh,
And I was just talking to Jeremy, well, question that they asked the ESA was, well, is any of this barcoded? Well, the answer is no. This was done when molecular work was just starting in Lepidoptera, in fact. And Manuel was the person who was involved in sending material to Regier's lab at the University of Maryland to start working on the first phylogeny of Lepidoptera. And it was just starting. And the other question that comes up is, why did I only just publish it last year? And the problem is that I did this field work right before I became research leader, and then I couldn't work on it for 10 years at least. So the priorities were phylogeny, the Lepidoptera phylogeny, particularly Pyrmoid phylogeny. So, but the specimens are still here. And that's the other question they asked. Do you plan on going back? And the answer, because of the narcos. And I said, no, absolutely, because of the narcos. So it was a good thing to go at that time because we can no longer go. Yeah.
Thanks. Kojin requested if he could go next. Kojin, can you hear me?
Oh, yep, I can hear you.
Go ahead and show me.
Okay. I'll go ahead and share my screen.
Does anyone recognize Kojin? He was a postdoctoral fellow. APHIS. Are you still on paternity leave, Kojin?
Yeah, kind of on mixed paternity leave right now. Yeah. But yeah, I was actually invited to give a talk back in, I think, May 2020, but we all know how that kind of went that year. So yeah, as Al mentioned, I was a postdoc at SEL from 2019 to last year, and currently I'm a biological scientist at the Pest Identification Technology Lab in Fort Collins, but based out of Alexandria, Virginia. So yeah, today I wanted to give a talk. So this talk I also gave at ESA in the SCL symposium that was held there. And it sort of describes the program that I run for APHIS S&T, which is the Identification Technology Program. And what we are is we're a program within APHIS S&T, Science and Technology, whose goal is to support state and federal programs in their mission to safeguard U.S. agriculture and natural resources against the entry, establishment, and spread of economically and environmentally significant pests. And we do this by delivering digital diagnostic resources for screening and identifying taxa that pose a risk to agriculture, natural resources, and our trading partners. So a lot of the folks who are there as part of SEL might be already familiar with our resources. But the projects that my program sort of operates, we operate these three main projects. One is called ID-AIDS, which is a searchable database of identification resources. We also maintain a very large image database called Image ID, which focuses on taxa intercepted at ports. And more kind of our conspicuous resources for public consumption are websites that we call ID tools, which are detailed websites that deal with identification of specific pestiferous taxonomic groups or pests of various commodities. Our program is supported by a robust staff of 14 federal and university cooperators. And we primarily work in conjunction with Colorado State University, where we have a lot of team members who are sort of in charge of project management and adding data to our systems. And we also have a lot of development and IT support for our digital resources from North Carolina State University and University of Georgia. We also primarily, so, you know, within the team of 14 of us, we only have 2 taxonomists on it, me and a botanist right now. So we are also extremely reliant upon the larger community of taxonomists, federal, state, and academic institutions. And many of the folks with ESW have collaborated and cooperated with us through the years. So now I kind of just want to walk through the 3 project areas again. So ID-AIDS, so this is a resource database that we maintain that has over 10,000 resources, including websites, extension documents, pest alerts, and also primary literature as well. And for each of the resources in this database, we capture a lot of metadata. So we record all the taxa covered in it. We also kind of record what kind of useful information is in it, whether there's a key or descriptions or distributional data. And there's also a fairly complex search algorithm to allow both taxonomic novices and as well as experts to find the resources they need for their identification needs. And this is really, yeah, even though it's applicable to all identifiers, it is especially useful for novices to certain groups who might not be familiar with available resources. And to sort of hello?
Oh.
Okay. And so sort of to expand this database, one of our main initiatives right now has been working very closely with the various national taxonomists within the Systematic Entomology Lab, within NIS, within other groups of APHIS, to incorporate primary literature that they consider to be really essential for sort of the identification workflow that a lot of the port identifiers and state identifiers and various screeners have. Moving on to Image ID. So this is our searchable database, which contains over 220,000, actually, I think it's close to 250,000 images now. and it focuses mostly on pestiferous taxa and taxa intercepted at ports. And in fact, over 100,000 of our images come directly from port interceptions. And then the rest are pulled from various public open access sources, such as digitized museum specimens. But for the specimens that are intercepted out of the images of specimens intercepted out of the ports, we do capture a lot of metadata for this as well. So, we include the commodity was found on, an interception number, and also sort of the whatever kind of geographic data we have. But of course, these origins are not always the most accurate because it's really hard to track exactly all the stops that commodity might have passed through. But unfortunately, because we include some of this metadata, as it stands now, this large image data set is only for PPQ and ARS use, but we're actively working on figuring out how to mask the sensitive data so we can make it available to the broader public. We also, in terms of just aiding the identification sort of workflow and continuum, we have various features to allow taxonomic experts to either verify and confirm identifications are correct, and also a comment section for really live updates to various taxonomy that might be necessary. But we also do maintain a public image database that contains thousands of images. It's hosted on University of Georgia's Bugwood website, if you're familiar with that. And this contains all the images that we generate as part of our sort of in-house imaging operations for Collins, and also associated with ID Tools websites, which I'll talk about shortly. In terms of current projects that our program is looking to collaborate on, is we recently expanded our in-house imaging lab to really kind of increase our capabilities and speed for generating images. So we're working on filling in the taxonomic gaps in our data set to include all recently intercepted species, but we're also just looking with various experts and various pest groups to collaborate on any imaging projects. And we've also been contacted recently about leveraging some of the data in our database to train image recognition models, because a lot of the images that we have are kind of indicative and representative of the diagnostic images being taken at the port, so serve as sort of an excellent test set for all the machine learning based image recognition tools that are being developed right now. And now finally, I want to touch on ID Tools for a second. So these are public websites, again, with detailed resources for identifying specific taxonomic groups containing pests or pests associated with specific commodities or other agriculturally important groups. And since I'm presenting this on Zoom this time, I can actually share larger versions of what this looked like. So this is an example of one of our tools that we've developed in conjunction with researchers at the University of Utah, called Exotic Bee ID. And these ID tool websites have really detailed information. You can find information on biology of the groups that are covered in the tool. For novices, there's detailed glossaries, often with linked images to help learn the morphology of the group that's being covered by the tool. We have most of our tools have these nice image galleries that allow for certain filtering. And also kind of a key component are these detailed fact sheets for the taxa that are covered by the tool. And finally, kind of the most sort of useful feature is most of these tools are centered around a Lucid-based multi-entry identification key. If you're interested in seeing what other tools we have, if you visit our website, idtools.org, you can see the full listing. And even though in this talk, I'm kind of focusing on the entomology aspect of our program, ITP deals with all pestiferous groups. So we have tools for plants, for fungal pathogens, and so on and so forth. Okay, I'm going to go back to my slides. Oops, oh no, sorry. Okay, so the creation though of these websites, it's all managed by our content management system that we call FSM, or Fact Sheet Manager. So taxonomic experts just need to populate the data they want to include, the images, sort of like the text they want to appear on the fact sheets. And then we have a just HTML wrapper that takes that data and creates the really pleasing user-friendly websites that we host. So it's really straightforward. All we need from taxonomists is the data that's locked away in everyone's brains. In terms of current developments, I just thought I would maybe show off some of the tools that we're planning on releasing in the next 3 months. Some of this has been done in conjunction with folks involved with ESW. But one of the tools that are coming out soon is this guide to bruchine seed beetles, which includes a key to all the genera of bruchines. And also importantly, it deals with this pestiferous group, Acanthocelides, which is a pest of pinto beans and such. Unfortunately, this genus is a kind of a garbage dump genus with 300 or so species. This is sort of the first digital treatment of the species groups. So we're really excited to get this out in about a month. We also, one of our first tools we released was this tool for Tortricia identification, which was released all over 10 years ago. And we're about to release a huge update to that tool, which includes about another 100 species. Exotic hornets are in the news again with the new Vespa species coming in the south. So in collaboration with Matt Buffington, we're about to get ready to release a identification guide that covers Vespa. This is another really impressive tool that we're getting ready to release on Batrocera fruit flies. And this matrix-based key keys out 842 species that are currently in Dacini. So all the species outside of those that are currently Afrotropical region, because that fauna has already been treated. And finally, we're also going to be releasing this tool on cerambycid beetles, focusing on keys to identify softwood boring genera in the US, and also commonly intercepted invasive genera. So it includes like the top 100 most commonly intercepted species. And this kind of gives a little preview. Increasingly, with diagnostics, we're relying upon molecular data as well. So we're currently going to be building molecular diagnostic interfaces as well to support molecular diagnostics. So that wraps up my talk. Just as a final summary, the program I manage, ITP, we deliver digital resources to aid pest identification and detection. We have our resource database, image database, and these targeted websites. We have a really robust staff and dev team, so we're constantly adding new data and improving user functionality. So if any taxonomists would like to collaborate or lend their expertise to our efforts, just feel free to reach out. And with that, I just want to acknowledge our ITP staff and take any questions.
Must be an interesting position, like to see all this different work that's going on. It's getting pretty broad with the USDA.
In theory, could this be used as just a scaffold for any group? Make like an ID?
Yep. So the reason that it's sort of pest focused right now is because a lot of these studies were funded off of PPA farm bill agreements, but Yeah, we like, we have somebody making like a tool on, non-pestiferous jewel beetles and things like that. So, yeah, it can be used for the development of any sort of public facing identification resource.
Cogent,
Mhm.
are there other projects on, coming up soon? Or in the works, or?
Yeah, so sort of, we have a old Spodoptera tool that we need to work on. And also, I think starting next, well, it was funded last year, but a Diprion piney sawfly tool that should be starting up as well. And we're also working on a huge update of all of our scolatines, so ambrosia and bark beetle tools as well, in conjunction with um, that research Anthony Cognato, Sarah Smith team at the University of Michigan.
Thank you.
Thank you.
Our next speaker is Stu McCamey. Everybody knows he's the world expert on leaf covers. Yeah.
The image of.
Yeah. I did try to make it full screen.
Yeah, maybe. You need it on this.
I would help that. That's your thing. This is my only slide because it's presented as the poster at the meeting. I'll guide you around here.
Yeah, can we do that and then scroll down as you need?
Yeah, I'll say scroll.
Sure.
Thank you for having me. As I said, this is the poster presented. And first I want to explain the title. Why do I call them raindrop tree hoppers? That's because most of them, as you'll see in the right column, if you scroll down, you can see more of the right column. They're green and light, and they mimic raindrops clean, like raindrops clean the stems after a rain. And there's been a couple times where I pitched actual raindrops, thinking they were these guys. But they lose some of that green when they dry and die. Why do I have them hyper-reverse? Well, as I started borrowing more and more specimens, I was finding more and more species. I thought when I began this that it was going to be a simple task. Only 9 genera and 4 monobasic, and only 43 species. I thought it would be pretty simple, but I realized that there are a lot more new species. And going from 43 known species to possibly 800 new ones, I call that hyper-diverse. Oh, and also go up to the top. You'll see so most are green, but you'll see also in the left-hand side of the top bar that there's some black species with bright yellow or white marks or bands to really advertise themselves. And you'd ask, why are those advertising themselves when the rest of them are trying their best to hide? I think it has to do with the host. Ferns are a rare host for any hoppers and probably insects in general. But I found these black and yellow ones on ground ferns and also the unfurling fronds of tree ferns. So I think they're obtaining or generating toxins from the fern. So to give you a little more background, these are in the subfamily Darnini, the tribe Darnini rather. which is very morphologically diverse. So a lot have extensions at the wing bases or further up on the pronotum, superhumal horns. And these do not have any of those. But it's not just the absence of horns that unite them. There have been 2 phylogenetic studies that have tried, both morphological and molecular, and both show these as the monophyletic group. So there are 2 main goals to my project. The first is to establish the genera, which is not clear at all. And the second is to describe all the species. So for the genera, there are 9 genera, as I said. Stahl in 1869 in the key described most of them. And he relied heavily on the four-winged venation, which is generally a good idea in tree hoppers. Generally, in other tree hoppers, four-winged venation is stable within the tribe and sometimes even within the subfamily. But in this, looking at all the specimens I had, they covered more variation than Saul was aware of, a lot more. For example, I recently described the species from Kentucky, of all places, and it was an isolated population. We now know it's more widespread than that, but that isolated population among the specimens and even on the 2 sides within one specimen, there was so much variation that it encompassed like four genera. So obviously those are not generic characters. You can scroll down a little bit. So what you see there in the key in the middle is Stahl's key, translated from Latin, updated terminology, and then adding the 2 genera that were described later, and then eliminating all the characters that I know vary within a species. And this is what you get. You can see most of the genera are unsupported. But really is a big deal what the generic limits are going to be, and which ones are going to remain valid, which ones are going to be synonymous. And to deal with this, I have a large data matrix with about 100 characters, and it's largely completed, but still has a lot of work to do. And I'll use that to generate a phylogenetic estimate, and hopefully the genera will fall out and also with good support. For the species, and also I want to mention, scroll up a little bit. Up, no, I need down, sorry. Look on the left and you'll see the nymphs of these tree hoppers. And you can see there's a lot of differences and possibly good indicators of what the genera are. But there is a problem. In most tree hoppers, well, in a lot of tree hoppers, the nymphs are laid, the eggs are laid in large clutches, and the nymphs grow up in aggregations, often tended by the mother down below, and they're often tended by ants, like their honeydew. So it's easy to find those because you just follow the ant trails and look for aggregations between tree hoppers. And it's very easy to associate males with females and nymphs. But in this group, in fact, in all the subfamily, the nymphs and adults are solitary and never tended by ants. And the nymphs are very cryptic. So they're rarely collected, and only a small handful have been associated with adults. So there's not enough data to use those info characters. But there are other characters, principally on the gross dimensions of the proportions of the head, and also the spines on the femur and tibia of all 3 thoracic segments. And I found a character on the wing that's all overlooked, here stable, and some other characters. So the species side, there are their own challenges. The principal one is synonyms. I know I have synonyms, and there are 3 reasons why I have them. First is that a lot of these species, morphospecies, I only have a male or female of. So I have to associate males and females. That will generate hopefully a lot of synonyms. So I have both genders represented in each one. And also variation within the species. Things like you see all the little yellow dots on that top one. That could be good, but in fact that disappears usually when they age. They don't all have it, but sometimes they have it. It disappears. So I have to I have to associate males and females, and also the variation of the species. That's one cognizant. The other is that I've had multiple interns working on helping me sort these morphospecies. So, for example, the subfamily was unknown from the Caribbean islands, but I have 3 specimens separated by 3 interns, all from Guadalupe. I'm looking at the specimen, it's clear the synonyms. The third source for synonyms is that because there are so many species, certainly more than I can keep in my head at once, I have to treat each large loan as its own entity when I separate them into multiple species. So any overlap in areas, I'm going to find more synonyms. Like Colombia, Ecuador, Brazil, they'll have a lot of synonyms there. And I can sort out a lot of those synonyms by using my data set, finding matching, all the matching. And but I also plan to use barcodes for these, so I can first do a neighbor joining tree to ferret out more synonyms, identical sequences, and that would also associate more males and females. And then I would use those sequences to generate another phylogenetic estimate to see if the genera I want to establish based on morphology are corroborated by what the molecules say. And that's my target. Thank you.
What are the pictures at the top? So pretty.
Yeah, those are the dorsal views. Let's go to the top. Those are dorsal views of some of the species, the small selection. And I want to thank you and Tina for taking all the things images, and you for compiling the plates I'll be using, and Tina for compiling helping me put up the posters. It was nice. Are there any questions? Yeah.
How do they orient on the plants?
They're usually in the petiole basins, down again on the stem. They are kind of facing downward, just like the raindrop would.
I've seen a lot of these obviously at their, dried adult points, and a lot of them look exactly the same to me. So are there plans to do any dissection?
Yeah, I will be doing dissection. In fact, for getting the barcodes, obviously it's a lot of dissections that I have to do. And in order to get barcodes, I'm going to put the abdomens inside the solution, and that will not only inspect the DNA, but clear the abdomen, which will save me a lot of time. Another question that are there any other questions? Well, when you haven't asked, I can think of. Good, scroll down a little bit. So that's good. Stu, why is that one, the big horns are different? How do you even know it's a raindrop preopera if there are so few associations? In fact, that one is not associated with any result. Well, first of all, I know and I'm describing the nymphs of New World genera, and I have lots of genera in every tribe in the New World, and that doesn't fit any of them. Furthermore, there's a similar species with the big interior horn that was reared by a living contact I had, and that adult was definitely raindrop. But that's all. I don't have no other questions.
Yeah.
Oh, actually. There we go. So make sure it's in the computer.
Okay, thanks, Elsa.
Yeah. Okay, should be good to go.
Okay. So I probably should have prefaced this slide with some of, I've been involved in a big project on the genus Anastrepa for the last dozen years. It was a big team of people, like Brian Wiegman's lab at NC State, colleagues at the Florida Department of Agriculture. Some of the molecular work was done at LSD. But anyway, we got Farm Bill money and we were able to make a really great collection of species from throughout the neotropics where Anastrepa occurs. But we couldn't get everything. So one species group within Anastrepa that we couldn't really deal with too much is the Curvicauda group, which used to be the genus Toxotropana until we showed that it really is just a group of derived Anastrepa species. So anyway, there's 8 recognized species, but except for the papaya fruit fly Curvicauda, it's not very well studied. There's clearly more species, but we get all these different singleton specimens and quite hard to determine whether they're all the same species or not, a lot of them. So, and I think one reason for this is they're thought to be mimics of Vespid wasps. And there's not like a whole lot of variation in the genitalia, which are usually pretty useful in other species. And they all feed on fruits of latex-bearing plants, Caricaceae, papayas, relatives, and what used to be the Asclepiadaceae. They all have these fruits with thick husks. Larvae feed on the seeds inside. Within this group, Blanchard described 5 species that were all associated with switchover.
Sorry about that. I was able to fix it last time, but I just rebooted it. There you go. You're on the second slide, right?
Yes. Thank you.
Sorry about that.
So anyway, Blanchard described these 5 species all from the same host plants, based mainly on body color patterns. So it's mainly whether they were mostly dark or more predominantly yellow, whether or not they had bands on the abdomen.
You see
Switch over there.
go ahead. Here's the tab. Let's see. Oh, oops. You are still on. Yes, so use these guys. Great, right?
Yeah. Sorry about that. There are other species of Anastrepa where body patterns are very useful. We've been able to corroborate that with molecular data as well. And even within the Curvicauda group, Curvicauda and other species, Littoralis, do have body markings that are very useful and again corroborated with molecular data. But for the rest of the group, we really didn't have much in the way of samples, just old pin specimens for the most part. So I was kind of very intrigued when I was contacted by people in New Zealand working in biocontrol. There's a species of Arabia in New Zealand that's a big pest. It's a vine that grows all over the place. So they've been looking for biocontrol agents. And they actually reared some anastrefa out of it. And they asked me, well, what are these things? So in Uruguay, they reared flies from fruits that, according to the current morphology, were 3 different anastrefa species. But when we looked at the CO1 and then looking at the nuclear genes, phylogeny we're working on, they all seem to be the same thing. And also the New Zealand folks did some mating studies, and what were supposed to be different species were interbreeding. The offspring were also varied. They weren't all swarmorphic. So, and we didn't see any differences in the wing pattern or in the genitalia. This is, I think, the CO1 tree. This part over here, this is all the ones from Arabia in Uruguay. So they're almost, a lot of them were identical, all very similar. And the same with the AHE tree. You can't really see it very well, but this part of the tree right here is one of the ones from Arabia. So that's pretty clear. These are really just all one species. And then to top it off, one of my colleagues was going through all the tetratoid types in some of the European museums, and he found this one that had been described as a platystomatid, and it, in fact, is the oldest name for this group. So all these species are now stemmed from some of the anastrefa nigritae.
Were these nuclear genes too? So those are things you like to deal with pros or alcohol?
Just in alcohol. If we're using that's called anchored hybrid enrichment. It's kind of like UCEs.
Yeah.
So that was what was in NC State, Brian's lab. So we have a big phylogeny trying to wrap up the manuscript. It's got 235 species in it. Probably to figure out how to do figures. Show it. Anyway, thank you to all the speakers tonight. We appreciate you guys. Back over to Don.
One more order of business. Andrew, you need to approach the front here. That's the ceremonial, not so ceremonial gavel to you. Jansen, President. And you get to close the meeting, should you choose. This is the case for the gavel. Maintained in Matt's office, is that true?
That's why I found it.
Okay.
Now the video is on if you want
Yeah.
to show the gavel in front of you.
Oh, yeah, you-
Very efficient.
Wait a minute, we're gonna pass the gavel in front, wait a minute.
Yeah.
Where is it?
I think, is it that camera?
Yeah.
No, it's this camera. Isn't it?
Oh, no, you're right. It's that camera. This guy.
Oh.
Perfect.
There you go.
So, are there any, do I move or does somebody else move?
You can close the
No,
meeting with that gavel as you wish.
All right, well, I will.
Before you close, though, I think we all should give Don a big round of applause.
Yeah.
I guess, right?
Yay.
Thank you.
Knocked it out of the park.
And if you can use the old parliamentary maneuver like I call this meeting adjourned, no objections, so.
All right, well then, if there are no objections, I will adjourn the meeting. Great, hearing none.
Yes.
Adjourned until 9.